In the vast, enigmatic wilderness that is the human gut, there thrives a remarkable habitant known as Escherichia coli, more commonly referred to as E. coli. This microorganism, a tiny yet formidable creature, usually calls the intricate ecosystem of our intestines its home, contributing to the rich tapestry of life within us in myriad, often unseen ways. But what do we have here? An extraordinary spectacle unfolds before our very eyes, a sight of such rarity that it demands our utmost attention. E. coli, venturing far beyond the confines of its customary abode, embarks on an audacious journey into uncharted territories. And what's this? A revelation indeed. Another colony of E. coli, flourishing against all odds in an environment that would typically spell doom for such organisms. The substance in question, streptomycin, a formidable antibiotic, presents a hostile landscape that few can navigate successfully.
Okay, thanks David Attenborough, but I think I've got it from here. Hey there people, welcome to what might be either the most exciting or the most questionable decision I have made as an aspiring biologist, editing E. coli bacteria at home using a CRISPR kit. That's right folks, while some people bake sourdough bread or brew their own beer in their free time, I've decided to dabble in a bit of genetic engineering. Why, you ask? Because who needs regular hobbies when you can play with the building blocks of life itself? Join me as I embark on this slightly mad scientist journey armed with nothing but my CRISPR kit, a questionable understanding of genetics, and an undying love for science. Will I create a modified strand of E. coli, or will I just end up with crippling food poisoning from ingesting the bacteria? Stay tuned to find out. So what really is happening here?
In the vast and intricate wilderness that is the microscopic.
Not now. Anyways, so as I was saying, streptomycin is an antibiotic that binds to the ribosome in E. coli, essentially killing them, since without the ribosomes, the bacteria can't make proteins. Think of it this way. Imagine there are two kingdoms at war with each other. The E. coli kingdom is under attack. Streptomycin sends some spies inside the enemy territory, and those spies destroy the water reservoir of the kingdom. Now the E. coli the E. coli kingdom won't die immediately, but due to the lack of water, eventually they will cease to exist, since they can't reproduce or grow. What I'm doing with my CRISPR kit is that I'm changing the genome of a protein called RPSL that allows the streptomycin to bind to the ribosome. Once I change a single base pair in the lysine amino acid at position number 43, it turns into 309, preventing the streptomycin from binding, meaning preventing the spies from destroying the water reservoir. So, how did I do it? Once you have put on the gloves, you need to next use this measuring tube to measure out exactly 150 ml of water and put it into a glass bottle. Next, you have to take your LB agar media or any other agar media and dump all of its contents into the tube. This weighs roughly about 37 grams. Once you have dumped it inside, you have to shake it vigorously. Make sure that everything is dissolved. Next step is to heat the water. Make sure it doesn't boil, but everything has to dissolve. It should be clear. So now I have dissolved all the agar in the water. Step is to put the liquid our agar in the petri dish, and you just have to cover the bottom barely. Don't pour too much. So this much should be enough. It's just covering the bottom. So now you just have to let them rest for maybe 1 hour or even overnight. It's better to keep it overnight. That's what I'm gonna do. And after that, I'm gonna keep it upside down in the refrigerator at 4 degrees Celsius. They have to be upside down so that the condensation doesn't fall onto the dishes. So now we're going to use our E. coli bacteria, which is this, and in order for it to be useful, we need to add 100 UL of sterile water. So how you're going to do that is using a pipette. Make sure that it is 100. So first you take the sterile water. So now that it's properly pre-wetted, we have taken 100 UL, and now we're gonna add it to our E. coli bacteria. We're gonna close it up and shake. We're gonna use this to streak out the bacteria to spread it all over the agar. Like this. So it has to be in these loops. We have to go in a crisscross pattern and change the angle after each pattern. And once you have done that, we have properly streaked the bacteria. You have to be sure that we wash this before putting it back, otherwise all of our material gets contaminated. So I think this is good enough. Now we have to let it dry and then put it back, put the lid back on and keep it upside down at room temperature till the bacteria grows for maybe 1 or 2 days. Bacteria grow, as you can see here. We can start mixing it with our bacterial transformation mix. So first what you need to do is you need to pipette 100 micrometer of the transformation mix. So we have taken 100 after pre-wetting it. We have taken 100 micrometer and we're putting it into a new microfuge tube. Now we have to take the bacteria. So this smells very bad, but so we're going to use this to make an inoculation loop, which is just a crisscross pattern, and we're going to mix that with the transformation mix. So I've taken some bacteria. Once you have mixed the bacteria and the transformational mix, if you're planning to do the experiment the next day, then store the solution at 4 degrees Celsius in the fridge. And if you're trying to do it right now, then get your DNA template, the Cas9 and gRNA protein, and add 100 microns of sterile water using a pipette. Mix it and check if it's yellow. If it's not, you have to contact Odin. There might be a problem. Next step is to add 10 microns from each of the tubes to the transformational mix. Once you do that, you need to keep it in the fridge for 30 minutes at 4 degrees Celsius. Till then, prepare your microcentrifuge tube with the LB agar by adding 250 microns of normal water in it. Once it's mixed, after 30 minutes, you're gonna take the mix we had and put it in lukewarm water for 30 seconds. This is called a heat shock, and this is to wake up the bacteria. You have to add 250 microns of the LB agar microcentrifuge tube water to the mix and let it sit for maybe 4 hours at the very least. And to increase the chance of success, leave it there for even a day or two. Once you're done waiting, you can pour all the contents of the tube inside the streptomycin agar plate and steak it out using an inoculation loop. And then all you have to do is wait. This is what a successful experiment looks like. Thanks for watching the video, guys. Be sure to subscribe to my channel and like this video. See you next time.